Hi, my name is Lindsay Wythe, and I will be presenting my research on the reduction of Salmonella infantis on skin-on bone-in chicken thighs by CPC application and the impact on the skin microbiome. Poultry producers in the United States process billions of pounds of poultry in a year. However, salmonellosis is a common risk with the consumption of poultry. As such, Salmonella enterica is a public health concern. Currently, poultry processors utilize acidifiers to mitigate foodborne pathogens such as Salmonella enterica through poultry processing. However, due to the acid tolerant response seen in certain Salmonella enterica serovars to organic and inorganic acids, alternative interventions need to be employed. One prevalent serovar, Salmonella infantis, which has been isolated in poultry feed, live chickens, and raw products, has led to significant outbreaks in the US. In the past few years, an emerging strain of Salmonella infantis have been reported to have enhanced biofilm formation, adhesion, and invasion into avian and mammalian host cells, with increased resistance to antibiotics and other antimicrobials. Therefore, the objectives of the current study were to evaluate the effects of CPC and PAA as short dips on the reduction of S. infantis on the microbiome and on the microbiota of the subsequent part rinsates. But overall hypothesis is that CPC will reduce the load of Salmonella infantis better than PAA on skin on poultry parts and alter the microbiota of the skin surface. For this study, 100 skin-on bone-in chicken thighs were inoculated with 64 micrograms per milliliter nalidixic acid-resistant Salmonella infantis and were allowed to attach for 60 minutes at 4 degrees Celsius. Following the attachment, thighs were treated as a 20-second dip by fully submerging the part in the treatment and agitating to allow full coverage. Thighs were then aseptically removed and allowed to drip remaining treatment off for 2 minutes. Half of the thighs were stored at 4 degrees Celsius for 24 hours. At the time of microbial analysis, either 0-hour or 24-hour, thighs were shaken in 400 milliliters of NBPW and discarded. One milliliter of the rinsate was saved back for microbiome analysis, and the rinsate was 1 to 10 serial diluted and spot plated on TSA and XLD augers. For molecular analysis, genomic DNA was extracted and sequenced using an aluminum MiSeq platform targeting the V4 region of 16S rDNA. Sequencing reads were filtered and aligned in QIIME2, where alpha and beta diversity metrics were analyzed, and ANCOM analysis was performed. Microdata was analyzed in RStudio using a mixed effect model on the log10 transform CFU counts. Trial was evaluated as the mixed effect, and treatment and time were evaluated as the main effects. Means were separated with Tukey's HSD. Significance across all of these analyses was set at p is less than or equal to 0.05. Microbiome data were analyzed in QIIME2 2020.2. The two trials were analyzed separately, with the first trial analyzing shifts over time, while trial 2 looked only at the 24-hour time point. Significance for all analyses were set at p is less than 0.05, and where appropriate, q is less than 0.05. Alpha diversity pairwise differences were performed using Kruskal-Wallis, and beta diversity pairwise differences were analyzed using PermaNova and PermDisc. The first objective of the current study was to determine the effects of CPC application versus PAA application on reducing the population levels of Salmonella infantis on chicken parts. The data presented on this slide was presented on a poster last year at the 2020 virtual PSA. The abstract was submitted by myself, Lindsay Wythe. The second objective of the current study was to determine the overall effects of these treatments on the skin microbiota. The rinsates from the first trial were used to examine if shifts occurred over time in response to treatment. The main effects of treatment and time and the interaction between were evaluated for alpha and beta diversity. An interaction between treatment and time was not significant across alpha or beta diversity. The main effect of treatment was significant for Shannon's diversity using Kruskal-Wallis, but there were no significant pairwise differences. Treatment and time were both significant Ooh, excuse me. Treatment and time were both significant for weighted unifrac and Jacquard distance. Although there was no interaction between treatment and time using Adonis, there was a main effect for both treatment and time. For Jacquard, Permanova and Perm disc were significant for the main effects of treatment. NINTC thigh samples were different from thighs from all other groups for Permanova. Using Perm disc, however, the dispersion of the data were significant between NINTC thighs and thighs from other groups for Jacquard. For weighted unifrac, Permanova was significant for the main effect, but Perm disc was not. NINTC thigh samples were different from thighs from all other groups for Permanova. Significant differences were not found between red samples and thighs of other treatment groups. Unlike Trial 1, Trial 2 focused specifically on the 24-hour time point to provide a more defined snapshot into the microbiota response. Treatment was significant for Pylo's evenness and Shannon's diversity. The microbiota on non-inoculated thighs, or NINTC, were different in Pylo's evenness and Shannon's diversity, or richness, from the microbiota on thighs from all other groups. The microbiota from NTC thighs were different in evenness and richness from the microbiota on thighs from all other treated thighs. There were no statistical differences in microbiota evenness and richness in thighs treated with tap water, CPC, or PAA. The main effective treatment was significant in Permanova for Jacquard's dissimilarity. Treatment was not significant for Perm disc for Jacquard. The microbiota on NINTC thighs were statistically different in Jacquard to the microbiota on NTC thighs, but were not different from the microbiota on thighs from all other groups. The microbiota on NTC thighs were significantly different from the microbiota on thighs treated with CPC or with tap water. There were no differences in Jacquard between microbiota on thighs treated with CPC, PAA, or TW, or tap water. Because the main effective treatment was not significant for Perm disc and Jacquard, dispersions in the data did not account for any of these differences. The main effective treatment was significant through Permanova and Perm disc for weighted unifrac. Through Permanova, the microbiota on NINTC thighs were different in weighted unifrac from the microbiota on NTC thighs and on thighs treated with PAA and tap water. The microbiota on thighs treated with CPC and those on the NINTC thighs were not different. The microbiota on NTC thighs were different from that on thighs treated with CPC or tap water, but not those treated with PAA. There were no significant differences in the diversity on thighs treated with tap water, PAA, or CPC. The only significant pairwise differences in Perm disc for weighted unifrac was between the microbiota of the NINTC thighs and the NTC thighs. The pairwise differences between NINTC thighs and thighs treated with PAA, and then the differences between NTC thighs and thighs treated with CPC were trending towards significance. Trial 1 ANCOM results revealed significant shifts due to treatment over time at both the phylum and order levels. All taxa reported at the phylum level were significantly different. The most abundant phyla on NINTC thighs were firmicutes, and the most abundant phyla on all other thighs were proteobacteria. There were also greater proportions of background taxa compared to proteobacteria on CPC thighs at both time points, including firmicutes and Bacteroides. At the order level for Trial 1, the only significantly different taxa was Campylobacteriales. The overall amount of Campylobacteriales OTU reads were numerically low, at less than 30 for each treatment group, but were still considered significantly different between treatment groups. Trial 2 ANCOM also revealed significant shifts at the phyla and order levels in microbiota due to treatment. Only 4 phyla were significantly different in relative abundances: Proteobacteria, Firmicutes, Bacteroides, and a previously unassigned taxonomy. Similar to Trial 1, NINTC thighs had the lowest relative abundance of Proteobacteria and the highest of Firmicutes. Thighs treated with CPC had similar relative abundances of Proteobacteria and Firmicutes as the NINTC thighs. For Trial 2 ANCOM, at the order level, all reported taxa were significantly different at the order level. Numerically, thighs treated with CPC had greater relative abundances of lactobacillus similar to the NINTC thighs. Thighs treated with CPC, PAA, and tap water had greater relative abundances of bacillus than the no treatment control thighs. These results showed a possible over time inhibitory effect of Salmonella infantis when treated with CPC versus a relatively static effects of Salmonella infantis on thighs treated with PAA. Thighs treated with CPC had greater relative diversities in OTUs of indigenous microbial taxa than the other inoculated thighs at both time points and both trials. Therefore, it is reasonable to conclude that CPC is an effective alternative to PAA for use as an antimicrobial and poultry processing capable of reducing Salmonella infantis without negatively impacting the microbiota. I would like to thank the University of Wisconsin Meat Science and Animal Biologics Discovery and the University of Arkansas, both for their support during this research and the publication process. I would also like to thank Safe Foods for their support during this project. And if you have any questions, I am online and ready and willing to take them. Thank you.